Okay, I think we can get started. So thank you to everyone who's joined so far. My name is Natasha, and I'm happy to be moderating this session once again of the IPDGC webinar series. Today, I have the absolute pleasure to be welcoming our speaker, Dr. Bryan Traynor. So just a little bit of an introduction to Dr. Traynor. He's a neurologist and senior investigator at the National Institute on Aging and an adjunct faculty at Johns Hopkins University. He's best known for his work aimed at understanding the genetic etiology of amyotrophic lateral sclerosis, so ALS, and frontotemporal dementia, FTD. And he led the international consortium that identified pathogenic repeat expansions in C9orf72 as a common cause of ALS and FTD. Thank you so much, Dr. Traynor, for taking the time to address the group. Really looking forward to your talk. And with that, I'll hand over to you.
Okay, well, thank you, Natasha. That was a nice introduction, and good day, everybody. Thank you for the invitation to provide you with this lecture, and I hope that you can see my slides and you can hear my voice here. So the title of my lecture this morning is The Clinical Utility of ALS and Frontotemporal Genetics. So what I'm going to try and do is give a sort of broad sweep about ALS and a little bit about FTD as well, to see how this might actually fit into your clinical practice and so forth. So these are my, if I can go to the next slide. There we go. These are my disclosures. The main relevant ones here are the patents on C9orf72 testing and diagnostic applications, just because I'm going to be talking about C9orf72 at a later date. And that's probably the most relevant one in that list. Okay, so it's actually quite sort of, I think, timely to be talking about ALS at this stage, because it was actually just over 150 years ago that Jean-Marie Charcot first described the disease. And of course, he did so when he was actually working at the Salpêtrière Hospital in Paris in France. And in fact, in France, the disease is still known as Charcot's malady, or more correctly, Maladie de Charcot. Of course, in England, it's known as motor neuron disease, and in America, it's known as ALS. So it goes by many different names, but unfortunately, it is kind of pretty uniform across the world, and I think we all recognize it as a distinct clinical entity, if not a collection of clinical entities and syndromes. So what am I going to talk about today? I've got a fairly large number of slides. I'm hoping I'm going to be able to get through it within the allotted time and leave some time for questions at the end. But we're going to start off with talking about how important is amyotrophic lateral sclerosis, or ALS, for the general community. Why is it important, in other words, that we consider this and even think about this in terms of our healthcare planning? The other issue is, what is the current state of genetics for ALS and frontotemporal dementia? In other words, where do we stand right at the moment? What is it that we know? What is it that we don't know and we need to continue to work on? I think that one area that's particularly close to my heart and particularly germane to this particular talk is, how does the genetics sort of unify ALS and frontotemporal dementia? How does it show us that these two very clinically disparate diseases are actually the same entity, clinical entity? And then I think that, we spend a lot of money on genetics. We spend quite a considerable time and effort collecting the samples and then generating the genetic data and analyzing it. And I think that, it's more than just finding the next gene to a certain extent. It's what does genetics tell us about the disease? And then, of course, the most relevant thing, I think, and the whole reason why we do this, because genetics does not exist in a vacuum, is how do we apply this genetics back to the bedside in order to help the patients who are actually still devastated to this day, 150 years after Charcot still described this disease. We can do in terms of disease modification, very, very little for our patients. And is there an opportunity here for us to change that paradigm and to actually shift it towards a situation where ALS is not considered as devastating a disease as it is today? Okay, so let's start off with this. The key question is, how important is ALS to the community? And of course, here I'm talking about the global community. Well, I think that, this is a nice paper that was done by Carissa Arthur in our lab, who is now up at Hopkins. And what Carissa did was she took all of the existing epidemiological data, in other words, what we know about the incidence of ALS in the world today, and she calculated the number of cases of ALS. How many cases are there across the entire world? And of course, that's an important number to know. But I thought that she did something even more interesting, which is she used the UN predictions, the United Nations predictions of population growth over the next 20 years to try and figure out, well, where are we going to be in 20 years? How many cases of ALS are there going to be in 20 years? And lo and behold, what she actually found was that there was actually going to be a nearly 400,000 cases of ALS alive in the world in 2040, which does not sound like a small number. I mean, we always think of ALS as a rare disease. And actually, I think that these numbers belie that, that it really is actually quite a burden on our society and on our healthcare services. The interesting thing about this, in fact, is that it represents an increase of 69% in the number of cases. Now, I think a very important point here is that that does not mean that the incidence is increasing. What it represents is that right now, today, we actually have a relatively young global population. And over the next 20 years, that population is obviously going to age. We've got this bolus coming up into middle age, into late age. And it is at that age, because remember, ALS is a late onset disease, that you're going to see those cases. And it's because of the aging of the population that we actually are going to see this increase, this fairly dramatic 69% increase in the incidence and number of cases of ALS. Now, this is actually a slide that I always make sure to put in when I'm talking to drug companies, because one of our roles at the NIH is, of course, to try and stimulate the pharmaceutical companies to have interest in our particular diseases and try and get them away from this notion that it's a rare disease. And the next point is, I think, very germane to that. So these numbers that you see on the screen here of 400,000 and an increase of 69%, they are actually based on the premise that the incidence, that the survival of ALS is about 3 years. Now, the point I make to pharmaceutical companies is, imagine your drug, that you come up with a drug and you're able to effectively give it to patients and provide it, get it through FDA and various other regulatory agencies. If you are able to increase and prolong the survival of an ALS patient by 1 year, just 1 year, you are going to increase the number of people that you can sell your drug to by 1/3. And it's usually at that time that you kind of see the pharmaceutical company kind of go, whoa, and they kind of realize, okay, this might actually be a fertile market for them. But I also think that it brings home to us that, because ALS is such a rapidly progressive and devastating disease, that feeds into this notion that it's rare. And actually, if it was like Parkinson's or Alzheimer's, where the patients live 10, 12, 20 years, we would have an awful lot more ALS cases, and it would be recognized as a much more bigger deal within the healthcare expenditures and how we structure our healthcare. So I think that kind of sets the scene for why we want to actually spend all of this money on ALS research and really think about it and how we can actually make a difference in the lives of these patients. Now, the next step is, well, what have we done? Where are we with ALS? What point did all of that money get us to? So in other words, what do we currently know about ALS? So this is a nice graph that was put together by Ruth Chia and Alan Renton. And I think that Ruth is actually, Dr. Chia is actually in the audience today. And what she did is she put together a timeline of ALS genetics with the familial genes on the top, and of course the sporadic genes, the more common sporadic or random form of disease on the lower part of this graph here. And what you can see is that really our story, our timeline starts back in 1993 with the discovery of the first gene, SOD1, by Dr. Bob Brown up at the Massachusetts General Hospital. And what I think you can appreciate is that it really was quite flat all the way along. There were a couple of genes that were discovered along the way, but none that really were contributing large percentages to our understanding. And then you get TDP43, but the reals and FUS, which were two important discoveries, but then the real sort of shoot up in terms of the percentage that's known is C9orf72, the discovery of the repeat expansion within the C9orf72 gene. The other thing to notice about this is that you can see that there really is sort of, the longer you go on in more recent times, there's really been an explosion in the number of genes that have been discovered. And I think that that represents the fact that genome technology, both in terms of genotyping and actual sequencing, has advanced rather dramatically over that time. And I think it's reflected in all of this, this here. Now, if you're to look at the bottom part, the sporadic, really, you can see that, you know, we haven't been as successful in terms of discovering it. I mean, I think that the biggest jump really was C9orf72 and a couple of others along the line. And in the end of the day, what we actually know is we know the genetic causes of about 2/3 of familial ALS and about 1/3 of sporadic ALS. And I think, you know, that's something that's pretty good. We can all congratulate ourselves. There are many different laboratories around the world that were working on this and making contributions to this. And I think that it's a testament to all of those efforts. On the other hand, what it does say is that there's still 1/3 of familial disease that we don't know the cause of. And I think that's something that needs to be worked out fairly quickly. And of course, there's 90% of sporadic cases where we just simply don't know what the underlying cause is. Is it genetic? Is it environmental? Is it gene environmental? Is it stochastic or random? We simply don't know at this stage. And I think that that's one area that a lot of attention and focus is going to be placed over the next number of years. I apologize, I have a picture of Alan here. I mentioned Dr. Chia earlier. This is Dr. Alan Renton, who also wrote one of these papers here, one of these review papers, and of course was instrumental in the discovery of C9orf72. Now, one of the interesting things about, you know, so that's kind of a nice little snapshot of where we stand right at the moment in terms of our knowledge of ALS genetics. But of course, ALS, finding the next gene, it's good and it's always important. But I think that it's not just about finding the next gene. It's more about trying to understand the mechanisms by which the disease, these different mutations and different genes within those genes give rise to the death of the motor neuron. What is it that gives rise to that actual neurodegeneration? And I think that, you know, we always say, well, increasingly say, well, a lot of the genes that we've been discovering more recently have been rarer and rarer and rarer, but each of them provides, despite the fact that they're only explaining a small number or a handful of patients, they actually provide us with a sort of a lynch point within the cell to better understand the underlying processes. This is something that Dr. Chia put together quite nicely. It's a link, sort of a representation of all of the chromosomes starting at chromosome 1 at the top, 2, 3, all the way around to chromosome X and Y. And she put in the different genes that were known about ALS at the time. This is back 2 years ago. And thankfully, I'm actually glad to say that even since then, this is out of date because more genes have been found. But then she started to look at the function of these different genes and say, well, okay, certain themes are actually emerging. So of course, the first one would be proteostasis, and you can see them labeled here with green. We got C9orf72, VCF, and they interact with other genes. So we see this emerging from the crowd and the multitude of genes that have been discovered. But then other ones start coming out, such as RNA metabolism. I think that's going to be already has been proven to be very important in the pathogenesis of ALS and indeed FTD. Autophagy is central here. Cytoskeletal organization. This is I think increasingly coming to the fore. And John Landers, who has really sort of led the charge on pointing out that cytoskeletal organization is important here. I think this is really good sort of, he pointed this out at an early stage in many years ago, and I think that his theory has really proven to be correct over the years, and increasingly so. There are other ones that I think are becoming less and less important. Vesicle trafficking, I think, probably not as important as others. Axonal transport goes into the cytoskeletal, so that's clearly important. DNA damage, maybe, it's possible that that's important. But then there are other areas that are listed here that really, really, do not look like, I don't think they've stood up to the test of time. So I'm not quite sure about mitochondrial function and how that feeds into ALS. And I can pretty much guarantee that angiogenesis has absolutely nothing to do with ALS. And I will stand by that. I think that that is one theory that is really not borne out the test of time. Now, I think that the next area is sort of brings us on quite nicely to, well, you're thinking of this more globally now, and you're thinking of the different pathways on a molecular level. What about on the clinical side of things? What does the genetics tell us about the clinical side of things? And so that's what really sort of the next section that I'm going to talk about is going to address. And really, maybe no surprise, certainly to those of you who've heard me talk before, but what I'm going to talk about here is really sort of relabel this as talking about the C9orf72 pathogenic repeat expansion. And like a good Irishman, I always like to tell a story. So this is basically going back to the beginning and describing how this basically the odyssey that gave rise to C9, and then at the end talking about some of the interesting little facts about it and why we think this is such an important gene in both ALS and FTD and neurodegeneration in general. So our story really begins back here at the basically 2 decades ago, and about 2006, with the publication of 2 papers, one by Vance et al., one by Marizia et al., where they had actually identified an area on the short arm of chromosome 9 as being relevant to ALS and frontotemporal dementia. And basically, those 2 studies, together with a couple of other studies, identified a region of about 7 million base pairs on the short arm of chromosome 9. It's shown here by these 2 dotted lines. Basically, that's the overlap, as you can see here. Now, 7 million base pairs is fairly sizable, contained about 100 genes, which, it's kind of the typical that you expect with family linkage studies. But of course, as I mentioned earlier, the genetic technology was improving. And I think that we took advantage of that because we actually did a genome-wide association study of ALS in Finland. Now, it turns out that it's a really good place to do a study of ALS in Finland for 2 very good reasons. Number one, it is a highly conserved population. They have gone through not 1, but 2 population bottlenecks over the course of their history in the last 7,000 years, which means that they're ideal for the identification of disease genes. And secondly, Finland has one of the highest rates of ALS in the world for reasons that will become quickly apparent. Namely, they carry both SOD1 D90A and C9orf72. So when we did that genome-wide association study, we were not expecting this, but what we found was a very strong association signal right here on chromosome 9. This is actually one of the very first true association signals that was identified within ALS. And one of the advantages of the genotyping chips that was used to do this GWAS is that the SNPs are located much, much closer together. So they're much more densely arranged across the genome, which allowed us to narrow the area of interest down from that 7 million base pairs down to a region of only 100,000 base pairs. And this was really exciting because it only contained 3 genes. We thought that we would have it very quickly after identifying that. It only took us 4 years afterwards to actually find the underlying mutation. And I emphasize this because, you know, knowing where the mutation is of course a very, very strong step forward. But what you really need to know in order to do all of the, make the mice and to do the cell work and to ultimately come up with the therapy, you need to know what the underlying mutation is. So it's good to know where the mutation is. It's a starting step, but what is really crucial is where is that mutation? And of course, there were a lot of labs around the world, very, very good labs, who are working on this. It wasn't just us. And none of them were showing, coming up with what was the underlying mutation. It was really becoming quite frustrating. So we said, well, okay, we're at the NIH, our funding structure is different. Maybe we can approach this in a different way. So what we did is we reached out to a lot of those different labs around the world, and we said, rather than compete with each other, why don't we try and collaborate? And that include John Hardy, Hugh Morris, Stuart Pickering-Brown, Peter Huting, Pentti Tienari up in Helsinki, who was crucial to this particular finding. And then we reached an agreement with another consortium led by Rosa Rademakers down the Mayo Clinic. What we said is, look, we have the sequencing facilities at the NIH. You give us your samples, we will sequence them, and then we push the data out and we'll all analyze it together. And from an early stage, we focused on this particular family here, which is a family from the south of Wales that was collected by this marvelous neurologist and dedicated neurologist. And I say that because he spent 10 years, he was at that time living in Cardiff, and he spent 10 years on the weekend, his weekends going down and collecting the DNA and phenotyping this particular family, which we call the Gwent1 kindred. And this was really the family that we focused our efforts on, and we tried a whole range of techniques, none of which were showing what the underlying mutation was. And that kind of told us, well, look, it's one of two things. It wasn't wasted effort because it told us it was one of two things. It was either the insert of a new piece of data, a new piece of DNA into that area, or the entire area was inverted. So what we did was we basically sequenced that area really, really deeply. So normally today, if we are doing a whole genome sequencing, what we do is we basically sequence it to about 30x coverage, which means that each base pair would have been sequenced 30 times. Actually, with this particular area, what we sequenced, we sequenced chromosome 9 down to 400x. So in other words, every single base pair was sequenced about 400 times, which is just like a massive overkill in terms of the amount of sequencing depth. But what was interesting about that was we identified 8 different variants within that 100,000 base pairs that we had previously identified. There were 8 variants that had never ever been described in human population polymorphism databases. And 6 of those were within a 30 base pair region. So I went in and I looked at it. This is actually a screenshot from that day. This is actually a screenshot of what I saw. And what you see here, all of the reads are represented by gray bars. And actually, if you were to scroll down, this would be down onto the next floor. So really, you scroll down and down and down. So, and then same on this side. And what we saw was this sort of distinctive pattern whereby we had a situation where the base pairs were sequenced 400 times, then went down to 2, and then back up to 400. And that's a pattern that we identified as indicating that there is a problem with the underlying structure of the DNA. And indeed, when we looked at it in more detail, it turned out that it was lying right here within this CCCCGG hexanucleotide motif. Right here, this is a sort of representation of the C9orf72 gene, right here at the beginning of the gene. what it ultimately, that was kind of the first clue as to what was actually going on, because we then got out a pen and paper and traced it back. And what we found was actually this individual carried more of those GGGCC hexanucleotide motifs than were present in the actual reference genome. And that was the first clue after 4 years as to what was actually going on. And it turned out that what it was it was actually this massive hexanucleotide repeat expansion, GGGCC, that was inserted into that spot. Normally, people have less than 23, and what we found with these patients is that they had between 700 and 1600 repeats, and sometimes even more than that. And what I particularly love about this paper is that we used really cutting edge technology in order to identify it. And then to confirm it, we went back about 100 years and we used FISH, where we had a probe against chromosome 9 to actually show on the location on the chromosome 9, the short arm of chromosome 9, exactly where it was. So I really love the fact that we had this sort of juxtaposition between really cutting edge and really kind of old and well-established technology. This gave rise to 2 papers that were published back to back in Neuron in 2011 at this stage. One was from us and the other was from Dr. Rosa Rademacher's group from the Mayo Clinic, who had independently found this particular finding as well. Now, since then, each paper has been cited well over 3,000 times, which is, I think you can imagine, quite a lot. But that gives rise to the question, well, why is it being cited so many times? Why is C9orf72 so important? Well, it turns out that there are a couple of reasons for this. First and foremost, it's really frequent. In fact, it turns out that it's one of the most common causes of neurodegeneration, not just in ALS and FTD, but across the gamut, possibly excluding ApoE. So overall, it accounts for about 40% of familial ALS and frontotemporal dementia. If you look at the sporadic side, it accounts for about 8% of sporadic ALS and frontotemporal cases. And when you add them together, because remember, 10% of ALS cases are familial and 90% are sporadic. When you add them together and do the math, it turns out that about 1 in 10 ALS and FTD cases are due to C9orf72. Excuse me. And this has really changed the approach to clinical approach to these patients, because now typically, no matter whether they come in with familial disease or sporadic disease, the first genetic test that's done is actually C9orf72. And actually the kind of the standard that's grown up, I don't know whether it's codified in any classification system, but I'm just saying what the actual typical approach of ALS and FTD neurologists is that they first and foremost screen for C9, and they will only spend the money on an exome or on doing the ALS genetic panel if the C9orf72 comes back as negative. And of course, it's a relatively cheap test to do. So it actually has led to sort of a sort of streamlining, so to speak, of ALS genetics. Now, the other reason why people are excited about this is because it literally explains every single case of overlap between ALS and FTD. Not every single case. There's a couple out there that are due to other diseases. We recently found one that's due to KIF5A, where you see ALS and FTD. But the vast majority of ALS, if you see a patient with both ALS and FTD, or a family where ALS and FTD is present, it is quite likely that C9orf72 is involved there. And of course, this is really interesting from an epidemiological perspective and from a diagnostic perspective. But I think that what's got people really excited is actually that this concept, and we knew this from the very first time that we identified this, that because it is a repeat expansion, it might be amenable to antisense oligos. And I'm really excited at the moment to see that Biogen has a trial ongoing in C9orf72. So hopefully we'll be able to shift that terrible curve, survival curve you see there on the right, over to the right a little bit, and hopefully give and offer some of these patients a little bit more hope than what is presently there. So we're really quite excited about that. Now, one of the things, and I think it's particularly germane, given that I'm giving a talk for the IPDGC Africa, is the geographical distribution of this particular C9orf72 mutation. What it turns out, and it's really quite interesting from a genetic and from a clinical perspective, is that it is very, very common in Caucasian European ancestry populations. It is diminishingly small outside of those populations. So you really see this sort of juxtaposition between Asia, for example, on one hand, and Europe and North America on the other. So it's really quite interesting that it's very common there, not so common in Asia and the Pacific Islands. We, of course, in ALS see the opposite as well. So optineurin, which was first described in Japan, in Japanese patients, it is known to be quite common among that particular population, and by contrast is not so common in European populations. So we see this geographical shift back and forth. It would be very interesting to work out how that is in Africa and in the Middle East and in other places as well. I don't think that that data is out there yet, but maybe somebody in the audience is pursuing this already. So I really look forward to seeing those results coming out. I think that one of the aspects of genetics that I particularly love is that you can actually try and understand where the mutation came from. So this is a graph showing what we call the Finnish haplotype, which is a sequence of SNPs that we identified first and foremost among the Finnish cases that were carrying this. And once we'd actually discovered the underlying mutation, the hexanucleotide repeat expansion, we asked a simple question, which is, well, what about, do other people in other countries also carry the Finnish haplotype? And it turns out that yes, the answer to that is yes, to a varying extent. So the blue ones here, I believe, are Americans. The orange ones here are Italians. These red ones, I'm blanking on it right now, but I think that they may be UK. But I know for a fact that that sole green line at the top is the one Japanese patient that we found carrying the C9orf72 at the beginning. And it turns out that literally every patient who carried the repeat expansion, which is shown here, the location of it is shown here with the vertical dotted line, carried to a certain extent the Finnish population risk haplotype. And what that suggested to us was that it had started on one occasion in Scandinavia and then traveled around. Of course, that gave rise to a question, well, how did it travel around? Well, we actually think that it was these gentlemen on their summer holidays brought it around the rest of Europe. And what is particularly interesting, the history buff in me particularly likes this, that if you look at the areas where the Vikings actually conquered during the Middle Ages, it corresponds to a certain extent to where we now see high incidences of C9orf72 along the coast and indeed in Sardinia and Sicily. So I think that that's kind of one of those interesting little side stories that comes out and emerges from the genetics. So that brings us on to, well, okay, that was a few years ago. What about more recent stuff? I mean, genetics has been advancing by leaps and bounds in terms of the capacity and the throughput and how we analyze the data. And I think that a very good example of that is a paper that John Landers and myself recently published in Neuron, where we, on our side, in our lab, we had performed a genome-wide association study that identified a signal within the gene on chromosome 12 known as KIF5A, which is a kinesin gene. It encodes one of the subunits for the kinesin, which is the antigrade axonal transport gene. Note that this was based on a large cohort, 20,000 cases and 60,000 controls. And that gives you a sense of, the size of the cohorts. I would also like to point out that, ALS being a relatively rare disease, despite what I said earlier, 20,000 cases is still an awful lot of cases. But if you were to compare that to the size of GWASs that are now being done in Parkinson's disease and in more common Alzheimer's disease, it really does pale in comparison. So I think that there is still room to go there in terms of the GWASs. And then, of course, the reason why I mentioned John in this is because he was doing a separate exome sequencing project. And basically, he identified also using that exome sequencing, KIF5A. So we really had two disparate techniques. One was genome-wide association studies and one was exome sequencing studies pointing at the same gene. And of course, we came together and decided to publish this together. Now, just to show you what this actual protein does, is it working there? Yes, I can see the video moving forward. So this is the kinesin protein here, walking down the neurofilament of the axon. And what you can see is that it's dragging this big vesicle behind it. And this bit up here at the top is what binds to the vesicle. And then the bit at the bottom is sort of like the feet that's walking down and the legs that's walking down the filament. Now, this is particularly relevant because KIF5A was actually already known, probably many of you in the audience knew this, already known to neurologists as a cause of hereditary spastic paraparesis and indeed also Charcot-Marie-Tooth. But the mutations on causing those two diseases tended to be in the motor binding domain, basically down in the feet. Whereas the ones that we were seeing in ALS were over here on the right towards the other end, the tail, in the cargo binding. And I think that this is a really good example of pleiotrophy where different mutations within the same gene give rise to different forms of neurodegeneration. And quite frankly, this is something that we are seeing time and time again. I mean, as neurologists, we're trained to put patients into different silos and characterize them according to their presenting symptoms. And I think that that, of course, has stood the test of time. But I think that we should also recognize that the walls between those silos are more porous than we previously thought. And really, genomics is showing us the ground truth there. It's showing us the ability to actually get at what is the molecular mechanism underlying this and how are these diseases related to each other. We've seen this with ALS and FTD. We see another example of this with KIF5A. There are other examples out there, VCF and VCP, and I'm going to show you another one at the very end as well. So this paper was published in Neuron. I think that, you know, the reasons why it got into Neuron, because it's another GWAS. People would say, well, it's another GWAS. But the reason it got in was because there's overall over 100,000 samples. The data was publicly available, represented a collaboration between literally every major ALS genetics group out there. And unusually for GWAS, we were able to go from the GWAS hit to the gene. And I think that that's a theme that's emerged with the ALS side of things, whereby we have relatively few hits on our GWAS, but we are able to get to the underlying gene, in contrast to Alzheimer's and Parkinson's disease, where they have, 90 and 100 different hits, but they don't know what the underlying gene is. And so they have to embark on this rabbit hole of systems biology and functional genomics, which is really quite difficult to do and quite challenging. So I do think that ALS kind of has the advantage in that regard, which is quite pleasing on our side of things, our side of the street. So being a neurologist who continues to see these patients, it sort of brings up the question, well, this is all great, but why are you not seeing patients in the clinic? I still see patients in the clinic. How are you going to use this to help the patients? And to be frank and to be honest, when I moved down to the NIH 15 years ago, I came down with a particular vision, and that was first and foremost to find the genes that were underlying this particular devastating form of neurodegeneration, but then ultimately to use that knowledge for gene therapy. And of course, the idea here is it's almost like the U.S. military. The U.S. Marines has a saying, you know, if you can see it, you can kill it. And the same thing applies with I think with ALS. If you can find the gene, you can probably fix it with gene therapy. And that has really been the mantra. And I think that we've been very successful over the years in finding these genes and developing up these cohorts and these technologies. And now over that 15 years, there has been a sort of in parallel, nothing to do with us, but in parallel, this development in gene therapy. But I think the time is now really ripe to do it. Of course, it's not just about gene therapy. I mean, one thing that really sometimes we kind of forget about it, and I consider myself in this boat as well, is that genetics isn't just about gene therapy. There's all sorts of different aspects in how gene therapy, how genetics can help us. It helps us with the diagnosis. It helps us with understanding the pathophysiology. It helps us, hopefully, in determine what the prognosis and what the subtype of ALS it is, as well as the treatment. So I don't want to say this is all about treatment. There's actually there are different areas in which genetics, it kind of enters into all of the spheres that are important for ALS care and our ALS patients. Now, I think that we all recognize that, but it's worth saying again, because sometimes we forget it, that starting with finding the gene is only the first step on a very, very long road to a therapy. And typically that costs $2 billion. You used to say $1 billion, but with inflation, it's gone up to $2 billion. And usually 10 to 15 years from gene discovery to first in humans. Now, so you know, there's a lot of work to be done once the gene is published and it's out there. Our job is to get that gene out there as quickly as possible, so people can start working on it, including academic and pharmaceutical companies. I do think there are one or two examples where we actually kind of get, you know, I like to say luck. I think luck favors the prepared mind, but every so often you do get lucky. And I think that this is a very good example of it. Again, this is a project that was spearheaded by Dr. Chia. And basically what we found was there's this form of juvenile ALS. So ALS may be rare, but juvenile ALS is really rare. But when we looked at 3 of these patients, what we found were de novo mutations within this gene, SPTLC1, which encodes one of the sphingolipid metabolism pathway enzymes. And we saw multiple occurrences of this. And of course, these we were able to show that they had these patients had abnormal plasma sphingolipids. Some of this is still being worked on, so please don't take this as ground truth. There are abnormalities in there, but we need to be a little bit cautious based on just 1 or 2 patients. I think that really for us, the bit that was really interesting about this was this particular gene had already been identified as a cause of hereditary sensory autonomic neuropathy. And within that field, the neurologists and researchers in that field had discovered that if you give these patients high doses of serine, that they actually improve neurologically. And we did that with our cell cultures. We were able to show that they actually improved in terms of their cell health. And we actually gave serine to one of our patients. And one aspect of this particular clinical picture that I've neglected to mention is that all of these 3 children had really, really low weight and failure to thrive. And as soon as we started her on serine, her weight improved, indicating that there was at least some improvement in the underlying metabolism. So we're very excited about this finding, and hopefully that you know, we'll be able to show over the longer term that there's a neurological benefit. Right now, we haven't seen a neurological benefit, but hopefully over the longer term, we will be able to see that. And I think stay tuned for that particular story. Disease modification, though, I mean, that's really the future. That's the light at the end of the tunnel. There are ongoing clinical trials. One was published in Antisense oligo for SOD1 in New England Journal earlier this year, which was showing dramatic improvement in patients, particularly with fast and aggressive forms of SOD1. As I mentioned earlier, there's an ongoing clinical trial for C9orf72. Neil Schneider at Columbia is giving antisense oligos against FUS in patients who carry FUS mutations. One thing that I should emphasize, of course, that these antisense, this is true precision medicine. They only apply to patients that carry those particular mutations that are involved, like SOD1, C9, and FUS. So that's really kind of what I am most excited when I'm asked by my patients which clinical trials am I most excited about. I think it's those three here that are the most, the ones that I keep my eye on an ongoing basis. This is an extension of this. This is just something that recently was published by our lab by Ramita Dewan, who basically found using genome sequencing a handful of FTD and ALS patients. It's a small number, but they carry definitive expansions in HTT, which is of course the same gene and same expansion that underlines Huntington's disease. So again, we see this pleiotropic nature in general of genetic mutations, where the silos and the walls between the silos are being broken down. And we do in fact see that you have mutations in these different neurodegeneration disease genes causing different forms of neurodegeneration. And of course, we were lucky enough through Columbia, Richard Hickman and Thor Stein up in Boston, they had 2 brains from these patients, and we were able to show that they actually had a distinct pathological form of the disease with no degeneration of the striatum. So this is really exciting. For us, as researchers trying to understand what the causes of the disease, but also for the patients, it's particularly exciting because, of course, there are trials out there of antisense oligos targeting the Huntington expansion. So I think that it's going to be a small number of patients, but hopefully in those patients we can make a significant difference for them. And of course, the question is, does that now mean that that clinicians and neurologists who see ALS and FTD patients in their clinic, in the ALS clinics and the FTD clinics, should we be screening them for Huntington because it's an actionable item, something that we don't routinely do now? Of course, if the patient goes to a Huntington clinic, they get screened for Huntington, but they don't get screened if they go to an ALS clinic and FTD clinic. But maybe this kind of shows us that that's something we should be considering. So I'm going to end here now with a couple of main points, take-home messages from this talk. I think that the first one is that ALS is perhaps not as rare as we think it is. It's an increasing, the number of cases and the burden to the healthcare and economies is going to increase over time. And by the way, one thing that I didn't mention is that when we looked at Africa, the number of increases within Africa was particularly notable because of the aging of the population within that continent and that population of that continent. We know the genes for about 2/3 of familial cases and about 1/3 of sporadic cases, which is really good, but there's more work to be done for sure. I think that it's clear that C9orf72 is a major ALS FTD gene. It's kind of altered the paradigm by how we try and diagnose our patients and how we screen for genes, and hopefully, ultimately, pretty soon, is going to alter how we actually treat these patients and approach them. KIF5A is a cause of familial and sporadic ALS, the point here is not so much that it's KIF5A, which I think is a really nice paper, but really sort of illustrating the size of the cohorts and the collaborative nature that's needed in order to achieve those results. SPTLC1 is potentially a treatable cause of juvenile ALS, going to be very, very rare, but something that, in those handful of patients, again, for a cheap nutritional supplementation, we could make a difference in the lives of those patients potentially. And I think that ultimately our future is going to be gene therapy, which is something I'm particularly excited about. So I'm going to pause there, and I'm happy to take any questions that anybody might have. I see Natasha coming up onto the screen, so I'll stay quiet now.
Thank you so much, Dr. Traynor, for that very informative talk. For me, it was quite interesting to hear about how the discovery of the C9orf72 mutation actually came about, and also that theory a bit about the Vikings and how that might be, how it might have spread around. I found that quite interesting. So there are a few questions. I think we have time for a couple of questions. So this question comes from Shaima. They say, thank you very much. They would like to ask if there is a genetic mutation that's linked to younger age of onset in sporadic ALS. I'm sorry, you're muted, Dr. Traynor.
Yeah, so that's a very good question. And it's funny that I get asked that, and I'll tell you who asked that question most often of me. It's actually pharmaceutical companies, because what they particularly want is they want a target that they can go after that would work across the gamut of ALS cases, not just in that sort of tiny sliver of cases, because then they can sell their medication quite appropriately and help to a wider range of ALS patients. I think that there are hints out there, and there are certainly publications out there. Is there one that gives rise? No, I don't see that. Now, the other piece of information that's relevant here, and I'm harking back to my epidemiology days, because I actually started out as an epidemiologist working on ALS epidemiology. It is important to recognize that familial ALS, so the average age of onset of sporadic cases is about 65 years of age. If you look at just the familial cases, the age of onset is about 55. It's about a decade earlier. So even though you can't distinguish clinically a familial case from a sporadic case, the age is shifted to the left. It's shifted to earlier. And actually, by extension, whenever I see a younger patient in my clinic, I'm there in the back of my mind, I'm always going, there's a gene here. There's a gene here. Even if they're telling me that they have no family history, I'm thinking de novo mutation that arose by chance in this person or something else. So I think it's a very important question. I think we're putting a lot of effort and time into it. I just don't have an answer. Sorry.
Thank you for the answer, nonetheless. Another question from Masharid. How does one interpret the ALS FTD relationship on the basis of the hypothesis, one gene, many phenotypes, or one phenotype, many genes?
Well, I think both apply, right? I mean, I think that's what genetics is showing us. And it's not just in the ALS and FTD sphere, it's across the gamut of neurology, and it's across the gamut of extra neurology, non-neurological diseases. I think they really, there are countless examples where that happens, where we see one gene giving rise to multiple phenotypes, and one, if you take, for example, ALS, there are multiple genes that we can describe. If you look at FTD, there are multiple genes that we can describe, giving rise to that. So I think that one of my aims here with this particular talk, so I encountered this problem or issue, shall we say, many, many times, that when we come out and we say mutations in this gene give rise to ALS, and it had already been described that mutations in that gene gave rise to another disease, everyone goes, no, those diseases aren't related. They can't be. You're wrong. And actually, we should really sort of move beyond that and realize that genomics is kind of showing us the ground truth. And as long as the genomic data is solid, I mean, it's not always solid. We have to, it's probabilities, we deal with probabilities. But as long as it's solid and we can replicate it, you know, just because those diseases are clinically disparate isn't enough of an excuse to say, no, that gene can't cause that, right? So it is one of the things I was trying to get across here, that the one gene to multi, and then one disease to multi genes. So both absolutely apply.
Yeah, absolutely. Makes sense. This is an interesting question. Someone was asking about how much does the Antisense oligo treatment cost? I'm guessing it's still in clinical trials.
Well, it's still in, yeah. So this is something that's, very close. We're working very hard here at the NIH. One of the advantages that we have with our funding. So I can actually answer that question a little bit, okay? So it turns out, so forget about ALS for 1 second, but there were instances where somebody, it was published in the New England Journal by Timothy Yu, where he found a patient. I'm blanking on the name. It was not N. Lauren. I'm just blanking on the name of the disease right in the second. But basically, he saw this patient, and he developed a antisense oligo against this particular gene. He knew what the underlying gene was. It took him 18 months to do it, and it took him $2 million, right? And what we are trying to do here at the NIH is we're trying to look at this and say, well, look, why did it take 18 months? And why did it cost $2 million? And what can we do to decrease that? So it turns out that it's two reasons. Number one is manufacturing, okay? Now, manufacturing is an engineering problem. So we'll leave that to the engineers, okay? The other reason why it takes so long is, frankly, you have to get it through the FDA. You have to get the FDA to agree to an IND before you put it into humans. And there, the issue is that they are looking for toxicity studies, and they are looking for animal toxicity studies. So and they take time and they take money. And one of the things that we would like to try and do is to try and decrease the number of animals that have to be tested. Sometimes they ask for 3 models. If we could go down to 2 or even 1 model and use other mechanisms to predict toxicity, that would speed things up and decrease the cost. And I think so whoever asked that question, that it's actually a really prescient question, right? And what we're trying to do is we're trying to come up with a cookbook that has no intellectual property associated with it, that any academic will be able to take and go, right, this is how we do this for a relatively low cost. Just to make another point, rare diseases, right, defined as less than 50 or 200,000 in Europe or in America. Each of them is individually rare, but cumulatively, there are massive numbers of patients. Well, I was doing some back of the envelope calculations, and within some limits, it goes from 88 million to 240 million people around the world alive with rare diseases. The vast majority, I'm not going to say all of them, but the vast majority of them with neurological diseases, or at least a neurological component to these multi-system diseases. And if you can think that, right now we know the genetic cause of about 1/3 of those, we've got an opportunity here. And I think that, when you hear these numbers being bandied about, it kind of your eyes kind of open up, go, wow, there's a real opportunity here to do things. And I think, and so there's a lot more. I could go on for an hour. I could do a whole separate talk about gene therapy. But this is what's particularly exciting at the moment. I think it's going to change. Honestly, if you're a medical student today, you're going to be practicing medicine totally different by the time you come to retire.
Absolutely. I think it's really exciting, actually, all these new innovations that are coming up, and because the technologies are also moving so rapidly. I think we have time for one last question before we wrap up from Matteo. He says that he gave a quick look to the HTT ALS FTD paper, and it seems the expansion occurred also in 10 healthy controls. How do you interpret this finding?
Right. So again, I mentioned it earlier, it's probabilities, right? So that's what we do as geneticists. We look at the incidence in cases, and we look at the incidence in controls, and we try and work out what the probability is. Remember that, and I actually think, was it Matthias? That was his name. I think Matthias put his finger on an issue here that is very germane to all genetics of late onset diseases. Right. Remember, we are dealing with late onset diseases. You are going to find controls, albeit normal individuals, that carry these things, right? The question is, there's 2 questions, follow-up questions. Number 1, were they ultimately destined to go on and develop it, right? Number 2, which is even more interesting, well, if they didn't, why didn't they? right? And by the way, just one point we were very cognizant of this fact, and Ramita, Dr. Dewan was very cognizant of this as well. We limited our analysis to full pathogenic mutations to get away from that. So we only dealt with 40 and above to say, right, these patients who carry these are fully pathogenic. We expect them to go on and get disease. So.
Thank you so much for the answers and for the talk. Very insightful. I think the attendees really appreciated it, and it will be also on our YouTube and Facebook. But yeah, thank you so much for taking the time to address us, and thank you to all the attendees. I think this will be the last talk of the webinar series for 2020. So thank you for those who have been with us for most of the year. We will see you back here next year for more talks. And with that, I'd like to wish everyone a great morning, afternoon, or evening, depending on where you're located.
Thank you.
Thank you. Bye.